So we are going to be discussing cellular respiration. Oof, that is rough looking. But remember how I said cellular respiration is probably one of the most important biological processes, right? It's because it's through cellular respiration that most organisms create energy, right? And this energy is important because it's used by organisms, by cells, for different activities, right? For our plants, it's what's used. Plants need energy for their growth and their development, for all those metabolic processes that they go through where they create those different products, all requires energy. As humans, when we move our muscles, right, that's going to require energy. When our brains and nervous system sends electrical impulses, that requires energy. So all of these activities that are found that a normal organism undertakes require energy, and we're getting energy primarily through this process. So because of it, this is why cellular respiration is so important. And really what the process is that we are taking sugar and we're turning it into energy. Right? That's really what all this is. We're taking sugar and turning it into energy, and that sugar is in the form of glucose. Right? And if you remember, a bunch of glucose molecules together are carbohydrates, so this is how we're taking carbohydrates, turning them into energy. And that energy, what's important is that energy is going to take the form of ATP. Right? And we've learned about ATP before. This is the energy. You know what makes this hard is because I'm a lefty too. Currency. Let's see if I can rest my hand. Nope, can't do that. So this is the energy currency of the cell. Right, so ATP, it stands for adenosine triphosphate. Right, so it's an adenosine molecule and 3 phosphate groups. Right, and when our cells need energy for one of those processes that I talked about, what they can do is they can break off one of these phosphates, break this off, and that's going to release energy. Right, so we will have ATP, and we break off a phosphate to get ADP, adenosine diphosphate, plus a phosphate group, plus energy. So that ATP, this energy currency of the cell, that is really what's being created. That's the most important product in this cellular respiration. All right, so the actual equation for this is we're going to take our sugar, our glucose, so that's C6H12O6, and that is one of our inputs, and we're going to use oxygen, and out of that we are going to get CO2 plus H2O plus ATP. And actually what we're getting is we've balanced this equation out. With one glucose molecule, we're going to use 6 oxygens. It's going to create 6 O2, 6 CO2, 6 carbon dioxides, 6 waters, and ideally 38 ATP. I say ideally because it doesn't always create 38 ATP. That's kind of the ideal max that this process can create. It's not always going to do that. But we're going to learn it as 38 ATP. Okay, so that's a little very broad overview of cellular respiration, at least the process, what's happening. And as we learned on Tuesday, there are 3 steps, or 3 main stages of cellular respiration. We have glycolysis. It's our first stage. We have, next comes the Krebs cycle. And then lastly, we have the electron transport chain. Right? So those are the 3 main steps of cellular respiration. We're going to go into each of these steps. We're going to go into each of these ones and allow you to have a little better understanding of everything that's going on. So let's move, and we're going to start with glycolysis. Oops, spelling that wrong. Hold on. Just pause because I spelled glycolysis wrong. Glycolysis. Okay, so this is the first step of cellular respiration. Right, and what's happening here is that we are starting with our glucose molecule. Right, we're starting with our glucose, and it's being lysed. It's going to be split in half, and it gets split into 2 pyruvate molecules. Right? It gets lysed, it gets split up into 2 pyruvates. So our glucose over here, it's our 6 carbon molecule, and these pyruvates are 3 carbon. Right? Now, in this process requires some energy, just to start, right? So it's going to require 2 ATP. But it creates, we'll say 2 ATP needed, but it's going to create 4 ATP. So we get a net in this process of 2 ATP. Right? That's our net in this process. And again, we want to keep track of ATP because if we go back, can I go back? Yeah. If you go back, ATP right here, that energy currency of the cell, that's what we're really concerned about in this process, right? This cellular respiration is taking that glucose molecule and ultimately turning it into ATP, which can then be used to power different processes. Right? So we want to keep track of our ATP throughout this process. And just in glycolysis, we created 2. Right? We needed 2, we created 4, and we have a net overall of 2 ATP gained. But that's not the only product of glycolysis, right? So we have our 2 ATP that we're netting. We've created 2 pyruvates now. But we also created another important compound, and that is NADH. Right, so NADH is very important because this is going to be used later on in the electron transport chain. And really, it's that third step, that electron transport chain, where the majority of our ATP is created. That's where 34 ATP are created. And it's this compound, this NADH, that is going to feed that chain, electrons. Right, so this is really important. So out of glycolysis, we also create 2 of these NADHs. Right, and they get formed from a molecule compound NAD+. And what happens to this, we get added during this glycolysis process, we add a hydrogen proton and 2 electrons, and that yields our NADH. Right, so in this first step of cellular respiration, in this glycolysis step, we are splitting our glucose molecule into 2 pyruvates. So we're going from a single 6-carbon molecule to 2 3-carbon molecules. As part of that, we are gaining a net of 2 ATP, and we're also creating 2 NADHs, which, like I said, are going to be important later on in that electron transport chain. So a couple things that we should know about glycolysis besides this is that it doesn't require oxygen, so you don't need oxygen for this step. And this is done in the cytoplasm, right? So this takes place in cytoplasm, right? So if we go now and we look at an example of a plant cell, right? We're going to do a rough plant cell. We have our cell wall there. We have our large central vacuole. We may have our nucleus down here. That's got our DNA, right? Command center of the cell. We could have little ribosomes in here. Maybe the endoplasmic reticulum. All different organelles that we learned about earlier. We're going to have our chloroplasts, right? Important organelles where photosynthesis takes place. They have those little pancake structures called thylakoids in there. Right, trying to draw them well. We have another organelle which is going to be very important later on for this. That's our mitochondria down here. But for this part of glycolysis, like we said, it takes place in the cytoplasm. So all these organelles within our plant cell are sitting in a fluid, and that fluid is the cytoplasm. Right, so that is just the fluid that makes up this cell in which all these organelles sit. And that is where glycolysis is taking place. Now, there are 2 routes that these products can go to after glycolysis, right? So we have our ATP, our NADH, and our pyruvates. So our pyruvates and our NADHs can take 2 routes after glycolysis. And that depends on if there is oxygen present, sufficient oxygen. Right? So if there is oxygen, if O2, then those products, the pyruvate and the NADHs, are going to go into the mitochondria. And they're actually going to go into that very interior part there called the matrix. I'll do another, in the next slide, I'll zoom in on that. Right, so if there is oxygen, our pyruvates and our NADHs are going to travel into the mitochondria, and the rest of cellular respiration is going to take place. If no oxygen, then the cell undergoes an anaerobic respiration. Right, and like I said Tuesday, this anaerobic respiration, we're not going to go deep into it, but just so you have an understanding of it, it occurs when there is not enough oxygen for regular cellular respiration to take place. Right? So if you think about yourself and exercising, right? If you're exercising very, very hard, you start breathing heavy, eventually, whatever you're doing, your muscles might start feeling sore. You might get that burning sensation in your muscles. What's happening is your muscles are requiring energy, so they're requiring that ATP. Your body's trying to produce it. But it's requiring so much that your body can't keep up through cellular respiration, right? You're using up all the oxygen before you can take in enough, and you just need more and more energy. So because of that, you're running out of O2. Your body can't go through cellular respiration, but it's still demanding energy. So you have this other type of respiration, anaerobic, where you don't need oxygen. The downside of this anaerobic respiration is that it's not as productive, right? It only produces 2 ATP, which if we compare that to our cellular respiration, right here we're producing 38 ATP throughout this process, but in our anaerobic respiration we are only producing 2, right? So not nearly as productive. The other downside is that there are byproducts that are produced in anaerobic respiration. In animals, it is lactic acid, right? And that's actually what's causing that burning sensation in your muscles. It's the buildup of lactic acid. In yeast and some microorganisms, their anaerobic respiration is a little bit different. Their byproduct is alcohol, right? So we've actually used that for our benefit, right? We've used that process of anaerobic respiration to create alcohol that we can then consume. So, like I said, you don't need to know the process of anaerobic respiration, but it's important. It's good that you have an idea of this is what happens, right? It's not just cellular respiration. There are these other processes that can go on as well. All right. So, but we are going to start with this first scenario, right? So we had glycolysis. We split our glucose molecule into 2 pyruvates. We have created 2 ATP net. We have our 2 NADHs. And now we have oxygen, so those products are going to travel into our mitochondria, and we're going to go through the rest of cellular respiration. So, step 2 is the Krebs cycle. There's actually a step before the Krebs cycle. It's kind of an intermediary step. It's a preparatory step. Some people include it in the Krebs cycle, but it's actually different, right? It's actually not, it's a little more distinct from the Krebs cycle. And that step is called pyruvate oxidation. Right, so in pyruvate oxidation, essentially what we're doing is we're preparing our pyruvates to go into the Krebs cycle. Right, so remember we have our pyruvates. They were created in glycolysis, and they were our 3-carbon compound. And in this pyruvate oxidation step, we are converting that pyruvate to a very, very important compound called acetyl coenzyme A. We're just going to call it acetyl CoA. Right, so this acetyl CoA is very, very important. It's a 2-carbon compound. So you see we had our 3 carbons, we now have a 2-carbon compound. What happened to that carbon? Well, it gets joined up with an oxygen, and we get CO2, right? So this process requires oxygen. That's one of the reasons why oxygen is needed in this, because it combines to these carbons that get kind of lost from these compounds and hooks up with, like I said, the carbons and oxygens hook up and they create carbon dioxide, and that gets exhaled or respired. Okay, so from this process we convert our pyruvate to acetyl coenzyme A, and in this process we also create another NADH, right? And it's really one NADH for each pyruvate. And because we have 2 pyruvates, eventually we are going to create 2 NADHs through this process. And 2 acetyl coenzyme A's. right? Because we had two pyruvates. So two acetyl-CoAs. And if we're really keeping track of everything, we're also creating two CO2s, right? Okay, so this is that pyruvate oxidation step. Like I said, some people include it in the Krebs cycle. It's not really part of the Krebs cycle, so I'm keeping it distinct. It's this preparatory step between glycolysis and the Krebs cycle where we're essentially preparing this pyruvate to enter the Krebs cycle. Pyruvate itself doesn't go through the Krebs cycle. It needs to be turned into acetyl coenzyme A, and then that acetyl CoA is going to enter Krebs. Excuse me. So there we go. There's our preparatory step, and now we're going to move on to our second main step of cellular respiration, which is the Krebs cycle. Right, and this is also called the citric acid cycle. Right? So this is our second big step of cellular respiration. We've had glycolysis. We split our glucose into two pyruvates. Our pyruvates then went through that pyruvate oxidation where they got turned into acetyl coenzyme A. And now that acetyl coenzyme A is ready to enter the Krebs cycle. So I'm going to, let's see, what's the best way to do this? We're just going to go into the cycle. So, like I said, we have acetyl coenzyme A, and this is going to enter this cycle. And the first step is that this acetyl coenzyme A gets combined with another compound called oxaloacetate, or oxaloacetic acid. Right, so this is the first step. This is a 4-carbon compound, and we already know our acetyl-CoA is 2 carbons. And from this combination, we get citric acid. Right? That's where this name comes from, right? It's Krebs cycle. I think it's because probably Krebs discovered it. But it's also called the citric acid cycle because this first product that is produced from the joining of acetyl coenzyme A and oxaloacetate is citric acid. Citric acid is a 6-carbon compound. Right? Now, from this step, there are a series of steps that eventually get us back to oxaloacetate. Right, so the citric acid gets turned into a different compound, and that gets turned into a different compound, and et cetera, all the way until we get back to this oxaloacetate. For this course, you don't need to know those other compounds, right? What you need to know is that acetyl-CoA combines with oxaloacetate, forming citric acid, and at the way end of this cycle, it turns back into oxaloacetate. You need to know that. You don't really need to know these intermediary steps. But what is important is during this process of citric acid turning back into oxaloacetate, a number of other products are produced. Most importantly are those NADHs, right? So as this is happening, we are getting NAD plus is getting turned into NADH. And so this is NAD plus is getting turned into NADH. All right, so this actually happens 3 times. We get NADHs that are made. Right? We also have, we see here citric acid is a 6-carbon compound, oxaloacetate is 4-carbon, so we need to lose those 2 carbons to get back here, and those get joined up with oxygen to form CO2, right? So actually 2 of those get formed up to form CO2. And then we also have the production of another compound called FADH2. And this compound is going to act very similarly to NADH. Right? And this is formed from FAD. Right? So this FADH is going to act very similar to NADH. And if you recall, as I said in the beginning, it's this NADH that is going to be used in the third and final step of cellular respiration, the electron transport chain. And it's through that electron transport chain where the majority of our ATP is created. Right? So lastly, one other product of this cycle is that this actually produces 2 ATP as well. Right? So this is going to, actually, technically, this is going to produce a single ATP. Now the reason I say 2 is because this cycle happens twice. Right? Remember, we have 2 of these acetyl coenzyme A's. Right? Because through glycolysis, our glucose molecule was split into 2 pyruvates, and then those 2 pyruvates, each one was split into acetyl coenzyme A. So we have 2 of these acetyl coenzyme A's, so each one is going to go through this cycle. So all these products get multiplied by 2. All right, which brings us to, I think, we're at a good spot here now where we can go back and look at what are all the products that we've created so far, right? So that's what we're going to do. We're going to look at products, and we'll label products before the electron transport chain. Right, so this is everything, for the most part, everything that's produced right before we enter the electron transport chain. Right, so in glycolysis, spell that correctly? Yeah, I don't know why that's so hard for me to spell. So in glycolysis, right, if you remember way back at the beginning of this, we had a net of 2 ATP, and we also produced 2 NADHs, right? We also produced pyruvates, but those pyruvates are then used. So we're not going to include them in our products. We're really kind of concerned with just our ATP and NADHs. And carbon dioxide, I'll include that. So that's glycolysis. We have our 2 ATPs, our 2 NADHs. In our pyruvate oxidation step, let me do that. Our pyruvate oxidation, if you recall during that step, where we're taking our pyruvate and we're converting it to acetyl coenzyme A, we had the production of a single NADH for each pyruvate, and since we had 2 of them, we have 2 NADHs that are produced. During each pyruvate oxidation, we have the production of a single carbon dioxide, but again, because we have 2 of them, we end up with 2 carbon dioxide molecules. Right, so that's glycolysis, that's pyruvate oxidation. And then during our Krebs cycle, we had the production of an ATP, and that's per cycle, and because we have 2 of those acetyl coenzyme A's, that happens twice. So we get 2 ATP. We also have 2 carbon dioxides per cycle times 2. That gives us 4 CO2s. We have 3 NADHs. Whoops, times 2. That's going to give us 6 NADHs. And then we had our 1 FADH2 times 2 for each cycle, and that gives us a total of 2 FADH2. Oops, here's my 2. 2. Okay, so all told, from this process, right, from glycolysis, pyruvate oxidation, and the Krebs cycle, we have, let's count it up, we've got 2 ATP here, another 2 here, so we have 4 ATP. We've got carbon dioxides, 2 here, 4 here, 6 CO2. What about our NADHs? We have 2 that were produced in glycolysis, 2 that were produced in pyruvate oxidation, 2, 4, and then 6 in the Krebs cycle. That gives us 10. 10 NADHs. And then we have 2 FADHs that are produced in the Krebs cycle. 2 DH2s. Right, so these are our products before we're getting to the electron transport chain, right? These are what we've made so far through glycolysis, pyruvate oxidation, and the Krebs cycle. And if you see, if we go back to our main equation of cellular respiration, all the way back here in the thing, we see that we have created all of our carbon dioxide, right? But we've only created 4 of those ATP, right? We've got 34 more that we need to create, right? That's a lot more. And like I said in the beginning, most of our ATP is created in the electron transport chain. So, to give you a little heads up of how that happens, for each NADH is going to create 3 ATP. Right? So each one of these NADHs is going to create 3 ATP. Each one of these FADHs is going to create 2 ATP. So if we multiply that together, right, we've got 10 of these NADHs. Each one's going to produce 3 ATP. 10 times 3 is 30. 30. And then with our FADHs, each one's going to produce 2. So 2 times 2 is 4. That gives us 34 ATP that can be produced. And we combine that with our 4 ATP that's produced in glycolysis and Krebs cycle, and that gives us our 38. Right? So that's how we get to that number, that 38 number. And what we're going to see at the end of this, with our last couple slides here, is how that 34 ATP is actually created in this electron transport chain. So, before I get to how that electron transport chain works, we should look at our plant cell anatomy again. Right, so here is our plant cell. We'll do them tiny up here. Vacuole, nucleus, DNA's in there. And remember, over here we had our mitochondria. Yeah. So this mitochondria, this organelle, this is where the Krebs cycle, that pyruvate oxidation, and the electron transport chain all takes place, all in this mitochondria. Right? Glycolysis, we said, was in the cytoplasm, in the fluid where all these organelles sit. But the rest of this process is taking place in this mitochondria, this very, very important organelle. So we're going to zoom in on that, and we're going to look at our mitochondria here. And it has an outer membrane, but it also has this inner membrane that's kind of squiggly like this. And this inner membrane, each of those little squiggles is called a crista, but combined, all of them, if we do it like that, multiple crista are called cristae. Right, so we have an outer membrane, we have an inner membrane that's made up of cristae. We have this compartment between the cristae and the outer membrane is called the outer compartment. And this interior space here is called the matrix. Right, and that is where our pyruvate oxidation and our Krebs cycle takes place. It takes place in the matrix. Now, our electron transport chain is going to occur at this boundary between the matrix and the outer membrane. It's going to occur right here at this membrane of the cristae, or the crista. And I'll zoom in on that. I'll show you that, how that's going to be working out. So before we get to that, we're here in our last step, electron transport chain. I'm going to go back to black. Right, so we're in our last step of cellular respiration, the electron transport chain. And what I'm going to do is we're going to look at the first step of this process, right? We're in electron transport chain. The first step of the electron transport chain is that our NADH gets broken down back into NAD plus the hydrogen proton and 2 electrons. So that's the first step of this electron transport chain. The last step is that those electrons combine with hydrogen, the hydrogen proton, and a single oxygen atom. So a single oxygen, it's often written as 1/2 O2 to create water. Right? That's where our water gets created in this process. So, first step, our NADH gets broken down into NAD plus, a hydrogen proton, and our 2 electrons. And really it's these electrons that are the important part of this process. If you remember from photosynthesis and the light reaction, those photons are energy from the light shining down. It excites the electrons in our chlorophyll, and then those electrons jump from molecule to molecule. Well, a similar thing is happening here, right? Our NADH gets broken down into NAD plus and a hydrogen proton and these 2 electrons. And these 2 electrons they're in a high energy state. And just like in photosynthesis and the light reaction, here in the electron transport chain in cellular respiration, these electrons start jumping from molecule to molecule. And you don't need to know the molecules. We'll just do molecule 1, molecule 2, there's a couple of them. They jump from molecule to molecule, and eventually they end up in this last step, right, where they combine with a hydrogen proton and oxygen to create H2O. But as they jump from molecule to molecule, they release energy. Right? They're releasing energy. And this energy is very important because this energy is used to pump hydrogen protons, these guys, from the matrix into the outer compartment. Right? So let's take a look at that process. Right here is going to be a zoomed up, if I can get this to work. Come on. There we go. Okay, so right up here, we'll do, remember, here's our mitochondria. It's the outer compartment. And then this inner membrane is the crista or the cristae, all the little squiggles. Inside of the cristae, that's the matrix. That's where the Krebs cycle is taking place. That's where all these NADHs are. And then between the cristae and this outer membrane is the outer compartment. Right, so we're going to zoom in on this. Right, and we have our outer membrane right here. We have a crista right here, and we're going to say this crista, it's got a double membrane. In here, this is our matrix. Right, so embedded in the crista, in this membrane here, are these protein complexes. And then there's also a very important protein complex called ATP synthase. Right, so in these protein complexes here, here's all of our NADH has all been created, floating around in here. And like I said, this NADH gets broken apart, and we have this release of hydrogen protons and electrons. And these electrons bounce around in these protein complexes here that are embedded in the crista. And they bounce from molecule to molecule. And as they bounce from molecule to molecule, they are releasing energy. And that energy is used to pump these hydrogen protons out here into this outer compartment. Right? So all these hydrogen protons get pumped out into this outer compartment. And eventually we have a buildup of these hydrogen protons, right? More so than within the matrix. And what happens is these hydrogen protons want to move back into the matrix, right? They want to even these things out. There's a buildup out here. They want to even things out in the matrix. So there is a pathway, there's a way for them to get back in, and that is through this ATP synthase protein. Right? So they get pumped out into the outer compartment. They want to make their way back into the matrix. The way they can do that is through ATP synthase. Now, as they do this, this is really important. What happens is this protein kind of acts like there's like a cylinder inside. It kind of acts like a motor or a grinder, and there is ADP within these proteins. And there are also phosphate groups. And as our hydrogen protons flow back through these ATP synthase protein, it turns that turbine, and that turning action actually forces these two things together. It smashes the ADP and the phosphate group together, and from that we get the creation of ATP. Right? The whole kind of goal of this cellular respiration is the creation of this. Right? And that's what's happening. So each of those NADHs, it breaks apart. Those electrons flow from molecule to molecule. You can kind of think of this as energy up here. Here's NADH. All the way over here is when it forms into water. So we have our electron up here that's got high energy, and then these are all the molecules that are in that protein complex. And as it goes from molecule to molecule, it's losing energy. It's releasing energy. Right? All this energy is getting released. And that energy is going to pump these hydrogen protons. So it pumps these protons from the matrix out into this outer compartment. And eventually we have this buildup of these hydrogen protons. They want to get back into the matrix, and they do so by flowing through the ATP synthase. And when they flow through that ATP synthase, it smushes together ADP and another phosphate, and we get the creation of ATP. All right, like I said, for each NADH compound, it's producing 3 ATP, and all that together combines to eventually create 34 ATP in this process, and that is cellular respiration.